Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rozan, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question answer window on your screen. After the presentation, I will ask our speaker your questions. Your questions will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Graziella Piras from 908 Devices.
Thank you so much, Leah, and thank you, Bioprocess International, for hosting this webinar today with 908 Devices. Hello, everyone, and thank you so much for joining us today for this webinar about cell media analysis, a critical piece of the bioprocessing puzzle. So let's look at the puzzle. So cell culture media analysis, does it really matter? We'll look at a few examples. Amino acid impact on cell growth, productivity, and protein quality is going to help us see how that's still very important. And we'll look at some innovative analytical and data analysis solutions. We'll then go to look at strategies for cell culture media optimization. And then we'll look at two great case studies from some of our collaborators and users. One will be on high throughput span media analysis for antibody process understanding. The next one is a brand new case study showing the building of a model for better nutrient utilization and process control. So developing and monitoring a process to achieve high productivity and desired CQA, we know there is several key pieces of that puzzle. The cell line, of course, is very important. We want to have the right cell line with the right genetic and phenotypic attributes. We also want to have the right process, the bioreactor process and parameters. And then, of course, at the center of it all, the cell culture medium, which provides all the nutrients to the cells and allow for those high titer, low COGS also is very important, and high protein quality attribute to develop really safe and efficient drugs. So why look at, why consider cell culture medium analysis? Does it really matter? And there are a lot of examples out in the literature, and I've just picked a few that I thought were really nice to illustrate the point here. So in this first one, publication from 2015, looking at protein quality and titer, and really showing that balancing glucose and amino acid is critical for cell growth, IgG titer, but also glycosylation profile. We can see on the left here that the authors obtain a fourfold higher titer by really balancing the nutrients. And they quote, faster cell growth, higher IVC, and IgG production was obtained as a result of balancing amino acids concentration in the cell culture medium and the glucose consumption rate. Interestingly, they also uncovered that the glycan profile was modulated by the amino acid and the glucose in the medium. The presence of undesired MAN5 glycan was linked to depletion of glutamine in the cell culture environment. In this next example, great example showing the influence of amino acid nutrients for sequence variants. This example is a control of misincorporation of serine in asparagine sites. We see on the left there that there's an increased misincorporation of serine into asparagine site, and that correlates with asparagine depletion. And in this paper from 2010, the authors were able to alleviate this misincorporation by modulating the level of asparagine feeding into the cells, and they showed this result up to a 2,000-liter bioreactor. Another example showing titer and protective effect of balancing amino acid, also in a CHO process. 5 amino acids here were shown to be depleted during the time course fed batch experiment, and we see this on the left, the 5 amino acid, asparagine, glutamine, serine, cysteine, and tyrosine. And what the authors show here was that they obtained a 60% increase in titer by balancing serine, cysteine, and tyrosine. The other 2 amino acids didn't have an impact. But very importantly, the balancing and nutrition of adding these amino acid to avoid the depletion avoided this high lactate production, and so protecting the cells against the negative effect of high lactate. Finally, in the last example that I picked up, this article from 2017 shows process intensification, which is becoming a very hot topic in our industry, developing cell culture media for high cell density perfusion process. The authors actually developed a high perfusion medium starting from an existing fed batch media. And we see on the left with the circles there, the volumetric productivity, and they needed to increase that volumetric and specific productivity to be able to go into a continuous process. And they achieved this by balancing 4 key amino acids, glutamine, asparagine, glutamate, and aspartic acid, as well as some key vitamins, which led to an increase in cell-specific productivity. Again, this balancing and optimization led to lower ammonium and lactate production, all beneficial to the process. So there's plenty of examples that show why cell culture analysis is so important. And of course, we know that amino acids have key roles in the cells. They're, of course, the building blocks of proteins, but beyond that, are involved in cell metabolism. Of course, glutamine being an essential energy source along with glucose, but also amino acids get involved into the TCA cycle and are just central to cell functioning. There are a lot of examples, again, in the literature, but there are 3 main aspects where amino acid can impact key CQAs. They can provide a protective effect against cell stress in the presence of those negative metabolites like lactate and ammonia, but they can also actually contribute to that cell stress. So it's very important to balance those amino acids like glutamine and alanine and serine. Threonine has been shown to protect also against cell stresses. Of course, amino acids will have a key impact on cell growth, viability, and productivity. And we may think only essential amino acids. Actually, all the amino acids can have a key role here. We know serine, in some cases, can increase titer and cell growth. Tyrosine depletion can impact specific productivity. And of course, this is all specific to your cell line and to your process. So it's important to do that cell analysis to find which amino acids need to be balanced and carefully monitored and replenished. And the final aspect here is the impact of the amino acids on protein quality, which are going to affect those CQAs. There are examples of impact on charge variants, sequence variants, and glycosylation. And some are listed here. So lysine and histidine can impact charge variant. We've talked about the misincorporation of serine into asparagine sites. We also see tyrosine into phenylalanine sites, and many other examples. So we need the right tools to be able to do this cell culture analysis. So let's look at the tools that are available. So today we have great tool to measure the cellular and the extracellular environment at the point of need, like shown on the left. So viability, viable cell density, cell size. Of course, those are all available through great technologies. The key metabolites, some essential metabolites like those lactate and ammonia that we talked about, a few amino acids, some sugars can be measured at the point of need, and of course, process parameters through online sensors. Amino acids and vitamins that are so important to the process can also be monitored, but usually it requires drawing samples and sending it out to a core lab or a contract lab to be analyzed through HPLC, which requires a specific skill set and usually takes time. So at 908 Devices, we have developed a key innovative technology to address this gap here in analyzing amino acid and vitamins at the point of need. The Rebel instrument is basically a miniaturized mass spectroscopy technology that allows for the analysis of amino acid and vitamins in a rapid, easy, and efficient manner. And we'll look at that a little bit more in details. The Rebel analyzer basically can sit right next to your bioreactor. It uses very small samples, less than 30 microliters, and it's going to give you data on about 30 analytes, and amino acid and water-soluble vitamins. It's very quick. Each sample is analyzed in less than 10 minutes, and it's very easy to operate, so no need for special skills. So it is an efficient and cost-effective process development tool. The panel of analytes that the Rebel will provide is shown here. So all amino acid, some key water-soluble vitamins, and some bioamines. So let's look for a minute at the kind of data that we can generate with the Rebel. The case study I'm going to show you basically provides an example of typical method accuracy and reproducibility that we obtain with the Rebel when measuring analytes in a typical chemically defined show media. And you will see that the Rebel provides accuracy and reproducibility. So we looked at the concentration of 17 analytes by spiking a known standard into a chemically defined medium, and we obtained accuracy plus or minus 20%. The reproducibility was very near 15%. So let's look at the data. Here is the experimental design for this assessment. So we used a chemically defined commercial media that we purchased from a vendor, and we don't have the formulation. So to be able to assess accuracy, we actually spiked in an amino acid standard at known concentration of 1 millimolar. This standard contains 17 amino acids, all at 1 millimolar, except for cysteine, which is at 0.5. So the standard was spiked in 3 samples of this medium, and then we also analyzed the medium without the standard as a control. And each of these were injected 4 times into the Rebel for a total of 12 replicates for each samples. And the data is shown here. So what we're looking at here, the blue bars are the percent recovery of the spike metabolites. Remember, this standard contains 17 amino acids. Of course, the medium contains all the other amino acids and many other components. But this allows us to assess accuracy as well, since we know that we spiked in 1 or 0.5 millimolar of these analytes. The expected values that we calculate are the control media plus 1 millimolar or 0.5 millimolar for cysteine. And then the observed values are the spike media samples, and recovery is the observed over expected. And you can see that the accuracy, which is basically shown by the percent of the blue bars compared to the expected dotted line, hovers around 93% to 118%. So we have an accuracy of plus or minus 18%. And then the percent RSDs shown at the bottom here shows us a little bit on the reproducibility, which was below 16%. So great. So now we can generate multivariate data from the Rebel. And like I said, each sample is analyzed very quickly, and we can run 96-volt plates. So we're going to generate a lot of data. Imagine you're running a microbioreactor, a time course, lots of data points, lots of repeats. That generates a lot of data, and you may want to use a data visualization tool. So how do we do that? How do we make sense of the data, which is super important? It's not enough to get the data. We need to be able to interpret it and make sense out of it. And for this, what we've done is the Rebel will generate a CSV file report that you can import into a data analysis or into a simple Excel, but also you can connect with the Sartorius Simca data analysis software, which is a great tool to do multivariate analysis of the data and will allow, it's an analysis tool that unlocks rapid data visualization and multivariate data trends, basically. So we can make sense of the data that's generated and be able to make those decisions that are so important when developing a process or running a process. So what can we do with this? Well, if you're looking at QCing your medium, making sure that the medium you get is what you expect, basically visualization tools allows you to do that. Media screening, if you're screening many different media from many different vendors, you want to make sure you're looking at a diverse set of media. So a tool like the SIMCA allows you to visualize that. If you're doing spend medium analysis and medium optimization, of course, you want to track trends during your fed batch time course and DOE and remedy depletion, make those decisions based on that data and remedy those depletion or overaccumulation of amino acids. And then finally, monitor your process, leverage metabolic profile to enhance, your process and enable PAT initiatives. So how do we optimize cell culture medium for your cell line? It is so important to really optimize your medium for each cell line and each clone. It is well known that using a single medium for many different cell lines doesn't really work. There's always some level of optimization that's required. So you may start with, like I said, initially screening your medium, making sure every lot of medium you use is consistent, and running the Rebel on your fresh media sample is very easy and very quick, and you can do that. Screening media and making sure you look at a diverse media set is also enabled with our technology. There are cell line specific commercial media that offer a wide variety of media, and you can leverage and make sure you look at a diverse set of media. Once you are testing the media and now digging into more optimization, performing spend medium analysis during a time course, and I can't emphasize that it's very important to look at as many time points as possible. If your process shows, a dip, for example, in cell viability or titer is not reaching the level desired, it's very important to monitor your amino acids and vitamin throughout that time course so that you know when to intervene and remedy the depletion or overaccumulation of amino acids. So as many data points as possible. And definitely a tool like the Rebel enables that very easily. You don't have to wait for your results for 2 weeks. You're going to see those results right away. And then remedy your depletion or overaccumulation and develop your feed strategy. And then finally use spend medium analysis to monitor cell health and make sure everything is going according to plan. So that medium optimization and monitoring is important throughout the process and needs to be done for each cell line or clone. Now, you may say, well, I'm early in my process development. I'm going to use a commercial media and feed. These are great. There's great media out there. They have been optimized. However, you still want to do that media QC. And if you're looking for the right media, remember how you can basically ensure you're screening a diverse set of media. You may choose to do some limited optimization, and a tool like the Rebel can really enable that because you have that data at your fingertip. If you have a proprietary platform medium and feed, again, that's maybe used as is for your early process development. But again, media QC and screening is always applicable. And then, of course, as you move towards your later process development, you will do more optimization, and the use of spend medium analysis and deep media optimization is going to become even more relevant. So at all stages of development, we want to make sure we leverage that cell media analysis. Let's look at media QC. So why did I mention this? Well, here's an example where we compared actually media composition of dry medium 199 with a liquid version of the medium. And what we found was that actually some analytes deviate from specification. So this is a classical media. We do have the formulation. And we actually showed that Some of these lots differ from the formulation by up to 50%. And we also saw differences between the liquid and the dry. You may want to be aware of this so that, if you see variability in your process later, when you switch from liquid to dry, usually used in later process development and definitely manufacturing, you're aware of these issues. Another medium QC experiment that we did was comparing fermentation media. So if you're developing a plasmid or an mRNA vaccine, you will be running E. coli and use some fermentation media. Fermentation media usually involves undefined components like hydrolysates, and these come with some variability. But what's striking here is that we compared LB and TB and other broths from different vendors and saw drastic differences between these vendors beyond what would be expected from a difference in undefined media. For example, in LB, we found, over twofold higher concentration of lysine and almost threefold higher concentration of threonine between the vendors. So yes, fermentation media can be variable, but it can be very inconsistent between vendors. So you may want to make sure you verify that. One more medium QC that's very important is if you are using FBS, and in some cases you may have to, you want to make sure that your analytics doesn't see any interference from the FBS. And what we show on the left here is we ran HO media and added different concentration of FBS and didn't see any interference in the analytics looking at the amino acid and vitamins. Also, no interference from having proteins, so no need for complicated precipitation steps. So at this point, I would like to walk you through two great case studies. And the first one is a high throughput spent medium analysis for antibody process understanding. So we talked a little bit about the negative effect of ammonia and lactate on cell culture process, right? We always want to make sure that we keep that ammonia level and that we see that switch in the lactate metabolism from that production of lactate glycolysis to actually lactate consumption later in the culture when the cells are starting to really crank out your antibody. There are other intermediates that actually are produced in the cell culture medium that are derived actually from amino acid metabolism. So this actually a reference to a great review published about 3 years ago here, showing how the fact that the cells are metabolizing amino acid and glucose and generating these intermediate and toxic metabolites, it's very important to control these, and especially amino acid need to be controlled to provide enough nutrients, but without leading to inhibitory effect. So it's a fine balance. Again, ammonia, we know, comes from the breakdown of glutamine and other amino acids, and it really affects your growth rate, titer, and protein glycosylation. So that needs to be controlled. The lactate inhibits cell growth. It induces apoptosis, can reduce productivity, and you want to see that shift early on from lactate production to lactate consumption. Some amino acids like phenylalanine, tyrosine, tryptophan, and others actually can also generate intermediates that can be inhibitory to cell growth. So it's so important to monitor all of this. So I'm going to walk you through a study that was done with one of our collaborators, and two great papers were published. The first one in 2020, and the second one was just published a few months ago. The experimental design here is cultures in the Amber 250 microbioreactors were supplemented with 10 or 30 millimolar ammonia to study how cells react to this high ammonia and trying to uncover how cells can detoxify and react to these conditions. So all the typical metabolites, lactate, glucose, ammonia, and all the parameters were measured with typical instrumentation. What was new here was that amino acids were measured daily with the REBEL and amino acid net production rate calculated based on this data. What the result will show is that there's a correlation of negative impact of high ammonia stress on productivity and cell growth, and the papers really dig into understanding, the underlying amino acid metabolism and eventually elucidate how cells detoxify through specific amino acid metabolism. So let's look at the data. Again, the experimental design is shown here. So Amber 250 runs, and we have some vessels that as controls, and then vessels, 2 duplicate vessels with the 10 millimolar ammonia, and then 2 duplicate vessels with the 30 millimolar ammonia. And again, like I mentioned before, all the typical parameters were measured, and then what was new was the REBEL daily amino acid monitoring. And some of the conventional data we can see here, viable cell density, ammonia, glucose, etc. And you quickly can see, of course, as expected, that in the high ammonia conditions, which are in the blue and the purple here, you see a drastic impact on viable cell density. We see, of course, ammonia spiking. We see a high, the cells are not consuming glucose anymore. A dip in cell viability very quickly in the culture, no switch to that lactate consumption, and of course, dismissal titer. So this is all very expected, of course, in these high ammonia conditions. Now, as the authors started to dig more into what was happening on the amino acid level, what we see here is that some amino acids behave similarly between the stressed and the non-stressed condition. So on the left, we have the amino acid over time, over the time course, so the concentrations of the relative change of these amino acids over time. On the left is the unstressed condition, and on the right side is the 30 millimolar stress condition. And you can see several amino acids, most amino acids actually behave very similarly. Glutamine and glutamic acid and glycine, methionine, they're all very similar. But what stands out is the alanine. We can see that in the stress condition, we can see the alanine relative change starting to go up. And in the second paper, they demonstrated a higher net production rate in the ammonia stress condition of alanine only, actually. And what they demonstrate in this paper with additional experimentation in looking at transcriptomics and other methods, a primary mechanism used by the cells to detoxify was to convert intracellular ammonia and pyruvate into alanine, actually. So direct conversion of ammonia into alanine and not a de novo alanine production. So great papers, again, to look at. And these studies were all enabled by many technologies, of course, microbioreactors and sensors, but also the REBEL, which enabled this fast look into the amino acid metabolism. So the last case study I'm going to go through right now is a brand new case study that I'm very excited to present to you today for the first time. This is a modeling and predictive tool and the development of a mathematical model to better predict and better feed amino acids. So let's step back for a second. So cell culture medium optimization strategies of yesterday, today, and tomorrow. So I've talked a little bit about these strategies, but again, a great schematic here from a nice review published in 2018. On the left, what we did yesterday and we're still doing today, experimental strategies to media formulation and optimization, right? So maybe doing, altering some components, doing some media blending, which are all in the category of experimental strategies, supported, of course, by DOE. Where we are, of course, seeing a lot of activity and will be the way of tomorrow, I'm sure, is these prediction of media composition and cell performance with minimal set of experiments. So how do we get there? How do we avoid, having to run, hundreds of experiments to optimize media and being able to get to the right point? Well, it's going to, model-based strategies are really going to help us there. And there is a lot of work being done right now, and it's been going on for a while already. But there is, statistical, artificial intelligence methodologies that are being used to help us here. Mechanistic, stoichiometric, and flux analysis. All these are used to create these models, have better prediction, and really develop digital twins of our process. And this is all going to allow us to fine-tune process and reduce the number of experiments we have to do, and at the same time have an optimal process. So cell culture medium optimization, anything left to do? Yes, definitely. The challenges of today, we still see impact of raw material variability and impurities on process performance, so that medium QC is so important. We want a faster, cheaper. We're on a quest to ever going faster and lower those cost of goods, but we need to maintain those high quality. So process intensification, is being heavily invested in to achieve that. And there's all these new modalities, right? We're well beyond traditional MABs. So bi and tri-specific MABs, which may not achieve those high titers yet. So we still need to optimize medium to achieve that. Viral vectors, of course, for gene therapy and vaccines. And then, we've seen the emergence of mRNA vaccines. And so back to fermentation, to support those mRNA vaccines, we need to create those DNA plasmids templates. And so can we improve fermentation medium, maybe with chemically defined solutions also? So the solutions, there's many, but some key ones here: advanced analytics, faster, smaller, easier, and real time. I think I showed you some of those solutions, and the Rebel definitely plays a great role here. But modeling and predictive tool, what I just talked about, that digital twin, so that we are not running those, we can decrease the number of experimentation and have a better representation of our process, which will lead to optimization of media and process, including for those new modalities. So I am very happy and honored here to be able to share with you some results from a project that came out of the AMBIC consortium. So the Advanced Mammalian Biomanufacturing Innovation Center sponsors great project in our industry. And I'm going to show you a few results from one of the projects here, which goal was to basically control amino acid addition and limit accumulation of those undesired metabolites that we just saw. And really, which leads also to lowering the cost of goods, which is so important as well, and have a better control, process control. So the solution that they adopted was to develop a mathematical model that is representative of the process and replace some experimentation with modeling, getting to that digital twin. You will see the results show that the model can finally predict how much amino acid to add without, overloading the cells unnecessarily. And the forecast algorithm can guide and facilitate feeding of essential amino acid in a CHO-fed batch culture. So what do we need to build such a model? The development of these predictive models rely, first of all, they lead to optimization of growth and productivity, like I mentioned before. They lead to better utilization of nutrient and enable process intensification through process control. So what do we need? Well, we need a great cell culture process model. We need genome scale or metabolic models. And we need the right analytics tool to feed into the model. And we need input from the actual cell culture system. So I'm going to walk you through the experiment that was run, which is an example of comparing basically a fed batch process derived from experimental designing a feeding process derived from an empirical and knowledge data versus this forecasted and model guided feeding. So in this experiment, the authors did shake flask experiment with a 30 milliliter total volume. The basal medium actually lacks 3 key amino acids, leucine, lysine, and valine. They use a typical fed batch that is similar between the empirical and the forecasted guided. And this feed also contains, all the component except for the leucine, lysine, and valine. These 3 amino acids are fed with a separate feed. And that's what's different between the empirical and the forecasted. The empirical leucine, lysine, and valine feeding were based on their previous knowledge of the process and the typical feeding that they would always do. And that feed was done every 24 hours starting on day 3 for the empirical. For the forecasted guided, they interrogated the model that they had built. And this model basically gave them, the amount of leucine, lysine, and valine that the model thinks should be optimal to achieve similar growth and titer. So let's look at the results. So the results are shown here. And what the conclusion was that the forecasted guided approach actually resulted in lower amino acid feeding while maintaining growth, and titer was almost as similar as in the empirical method. So we see on the top graph here the example of leucine. So leucine was one of those amino acids that was not present in the feed, but was fed based either on the empirical or forecasted guided approach. And the empirical, you can see, starts pretty high and then over time goes down. The forecasted guide actually, told them to feed at a minimal level, which was much lower than the empirical. So right away, you can imagine a better control on the feeding here. The result actually, very interestingly, is that the growth was similar. The viable cell numbers were identical between the two. The titer result shown on the bottom right here, we see that the forecasted guide showed slightly lower titer at the end of the process. And this actually model is still being developed as we speak. The author also think that when they looked at the other amino acid, they saw a slight difference between the two processes. And these differences and the difference in titer may be explained by the metabolic shift and mTOR signaling difference between these two processes. This work is, again, being published by the group here, sponsored by AMBIC, and will be coming out very soon. And the model here is also being still worked on. So embarking on predictive digital models for cell culture metabolism. So previously, glucose and dissolved oxygen and pH, temperature, and sometimes, also other metabolites easily measured. But what has been missing as a at-line or online measurement are those key nutrients. And 908 Devices has done, a tremendous amount of work to basically allow that point of need analysis there for the amino acids and vitamins by developing the Rebel, which is the instrument that I've been talking about. And all of this together is going to allow the building of these models that are so important for direct multivariate insight into the process and enriching the predictability of how to feed the amino acid to have that right balance of feeding without, leading to the overaccumulation that leads to those negative metabolites. And as I talked a little bit about process intensification as well, it's so important that those media are fully optimized for perfusion process. As you all know, that a lot of medium is used in perfusion. It's key to adjust and really have the right balancing there. So we have a lot of opportunities ahead of us for deeper insight into critical process parameters and process intensification and modeling. So cell culture media, does it matter? Yes, it matters, and it matters, still as much and if not more. So many ways: process intensification, renewed interest in fermentation, with renewed interest in producing from E. coli with these mRNA vaccines and other vaccines and applications, the impact on glycosylation and protein quality attributes. I haven't talked about a lot of these other new modalities like CAR-T and gene therapy. Maybe in a future webinar, we'll be able to elaborate more, but of course, lots of work being done there as well. And so much we need to learn about the influence of the cell culture medium to achieve those high quality attributes for those applications as well. So I think I've reached the end here. So I really would like to thank everyone for attending today, and I would be happy to answer a few questions. So thank you so much.
Okay, so it looks like we have a couple of questions. So the first question is, where can I access the modeling and predictive tool that you presented today?
Great question. Thank you so much for the question. So there's a couple of tools here that I talked about. The first one was the tool provided by Sartorius. So if you remember, I talked about how the data coming out of the REBEL can be directly connected to the SIMCA multivariate tool. That's one, so through Sartorius. The second one, which is more of the modeling tool that is being developed, that is through AMBIC, which is the project has been sponsored by AMBIC. So AMBIC is the consortium that I mentioned, and any member of the AMBIC consortia will have access to that great graphical user interface, basically. So you don't need to be an expert in modeling. You'll be able to use that tool, to apply it to your own process.
Do the samples need to be cell-free?
So yeah, that's the only, I would say, processing of the samples. So when you draw samples from your bioreactor, you want to spin it down or filter it, so you remove all those cells and cell debris, basically. But that's the only processing really that is needed to run on the REBEL. They can go straight into the REBEL, which has the liquid handler and everything to process your samples and give you that rich data.
Okay. Is there a benefit in performing cell media analysis for a perfusion process?
Definitely, I would say absolutely. As in, the goal of course, in a perfusion process is to really increase that volumetric productivity and cell-specific productivity. So you want to really optimize that medium. Also use a lot of medium, as you usually go through at least 1 or 2 volume exchange per day. That's a lot of medium, so you don't want to waste. It can become a big impact on your cost of goods. So you really want to adjust that, the nutrient concentration in that perfusion process. Also, if you're working on a fed batch and moving towards a perfusion process, but you want to sort of adapt your medium, you're going to have to optimize it really to achieve those high viable cell density. But also, you want to make sure that your cells are in that production mode and not, only, in that growth mode. So very important to adjust your nutrients in a perfusion process as well.
Okay. Should I measure amino acids daily or just at the start and the end of the culture?
It is very important to, that's a great question, actually. It's very important to measure, I would say daily. If we see some users or some user or process scientists measuring towards the end of the culture, thinking that's where viability is dropping, so let's look at it. But in reality, if you want to avoid that drop in viability, or if you want to remedy that low titer, you want to act before it happens, right? You want to make sure your cells are getting the nutrients in advance of that drop. When you see the drop in viability or that low titer, it's sort of too late. So you want to really profile your entire time course so that you can figure out when to remedy that depletion or overaccumulation. So the more data, the better. Why not even measure it several times a day? Maybe you can learn more from that too. And definitely with an instrument like the Rebel, which only requires a few microliters and is going to give you the data right away, that becomes very doable.
Do amino acids matter also in viral vector productivity and quality?
Yes, that's a very great question, actually. So it's still not as well characterized. I would say how to improve, a viral vector production titer. Of course, with viral vectors, one of the key question is how do I increase my full capsids? But also which is going to contribute to your productivity. But also, we see the industry needs higher, titers, basically. And it's a shorter process. A lot of process are transient transfections, but the medium, provides a nutrient. And there are already reports, we can see reports where key amino acids are, going to influence also that productivity. So I would say absolutely matters as well.
And can I measure all amino acids from one small sample with the Rebel?
Yeah, so the panel of 33 analytes provided for each measurement. So every time you run a sample, you will get that rich multivariate data off of the Rebel, basically. Yeah.
And can the Rebel be implemented into a GMP compliant environment?
Absolutely. So it is CFR 11 compliant, and absolutely can be. And we see some users already implemented that.
Okay, it looks like we have time for just one more question. If we didn't get to your question, we'll be passing those along to Graziella. So go ahead and continue to type your questions in. But the last question for the webcast is, are commercially available medium and feed not already optimized for CHO or other cell lines?
Right. So I did mention there's a lot of great commercially available media for CHO and other cell lines. So yes, they are. However, if you think about, how these media were developed, they were developed probably on a few cell lines specific to that vendor. So although, yes, some media out there and feeds also are great and have been optimized, there's always a level of optimization that is needed for your own cell line, for your own clone. At the end of the day, when you derive that cell line, that clone, you create a specific genomic and phenotypic environment, and those cells are may have different requirement. So yes, there are great media out there, but optimization is, especially if you're starting to build in your late phase development, optimization is absolutely needed, and monitoring is always needed to make sure everything is going according to plan.
So thank you, and thanks to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website, and as a registered attendee, we'll be sending you an email with a follow-up link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox.